My name is Missy Weiss. I am the Director of Citizen Science and Education with the Ocean Research and Conservation Association, or ORCA, and I'm also the project coordinator for this citizen science project. So for those of you who are not familiar with ORCA as an organization, we are a nonprofit. We were founded in 2005, and our mission is to protect and restore aquatic waterways and the species those waterways sustain. And we do that in four major ways. We do that through our innovative technology, or our Kilroys, which are real-time water monitoring devices. We currently have 25 of those real-time water monitoring devices throughout the Indian River Lagoon and in those freshwater canals entering into the waterways. And you can access that information at any time on our website. So our innovative technology, our real-time water monitoring devices are certainly the pillar and what ORC was initially founded on. We also uphold our mission through our applied science research. And under that, we have two umbrellas. We have an ecotoxicity as well as a One Health theme for our applied science research. We have our citizen science, which is what this presentation is really talking about today, which is one of our projects. And we have our education. And so our education is an opportunity to engage students from K all the way up into undergraduates and graduate students in real world science research. And so when it comes to our research, we really focus most of our efforts on the Indian River Lagoon, given its tremendous economic and ecological importance to our area. It encompasses 40% of Florida's East Coast. So clearly it impacts a lot of people and brings billions of dollars to our region. And so its environmental health is incredibly important to our area. With regards to our citizen science work, we currently have 4 citizen science projects that we invite people of all ages, backgrounds, ethnicities in the ability to participate in. And that includes our land to sea monitoring. This is composed of a buffered shoreline initiative as well as a living shoreline initiative. We have our pollution mapping citizen science project, where teams go out 4 times a year to collect water quality, water and sediment samples, and then help us analyze those samples for a panel of different analyses. We have our One Health fish monitoring project, which is the topic of today's presentation, as well as our student-centered citizen science project called A Day in the Life of the Indian River Lagoon, and now A Day in the Life of Lake Worth Lagoon. So we have expanded that project to actually go into another waterway a little bit further south than the lagoon. And that's really a way for students to enter into the world of citizen science, enter into the world of science research. And on a single day in October, we have over 2,000 people collected the same information on the same day about their adopted sampling location. So today's presentation is really about the One Health fish monitoring project. And so we have to really understand what the term One Health means. And One Health is an approach, it's an idea that the health of people is directly connected to the health of animals and our shared environment. So for example, when the environment's health is impacted through contamination or pollution, it can have direct impacts on human and animal health and can lead to disease and other health concerns. But what it also means is that when we protect one of these sectors, we actually indeed protect all of them. What One Health also recognizes is that it's going to require collaborative efforts of multiple disciplines working locally and globally to attain optimal health for people, animals, and our environment. And so for ORCA, what that really means is that we are responsible for collecting the needed data, the needed information, the unbiased information, or the evidence to help drive change. And then we also take it upon ourselves to make sure that we are sharing that with the other sectors involved in that collaborative effort. So whether that's the public health sector, the veterinary world, or even other science sectors. And so when we're specifically talking about our ORCA's One Health fish monitoring project, what we're really asking ourselves is, what are the pollutants entering into the Indian River Lagoon and freshwater ways connected to the lagoon? Are those pollutants accumulating into fish, and not just any fish, but fish that have been identified from our angler surveys as being fish that are commonly consumed by people? And then with that data, are those accumulated pollutants at a level that is potentially harming human health? So what are the potential human health risks, if any, through the consumption of those fish? And so with this data set, we envision sharing this information with those various sectors to help allow community members to make informed, educated decisions, but also be able to provide this information to local decision makers or stakeholders. And so at this time, we are testing for the following pollutants. And this list is always growing, or in some cases, some analytes are being removed depending on what we're finding. And so at this time, our fish samples are being tested for the biotoxin microcystin. This is a toxin that is produced by Microcystis, which is a cyanobacteria, and it has known human and animal health impacts. We also test all of our fish samples for the heavy metal total mercury, and in the past we have tested for lead and cadmium. We test nearly all of our fish samples for the herbicide glyphosate. So glyphosate is the chemical compound in products such as Roundup, but also found in other herbicides as well. We test for microplastics. And then if you attended the last findings to date, I shared our recent findings on per and polyfluoroalkyl substances, also known as PFAS or forever chemicals, in which we were able to obtain that data from our partners at the University of Florida. And so if you're interested in that information, you can access the last findings to date, or we can discuss it at the end of this presentation. And we're currently trying to develop methods in conjunction with Embry-Riddle Aeronautical University to detect for UV filters. And these are the chemical compounds found in some sunscreens. And so we know that there are UV filters entering into our waterways, but are those UV filters also entering into fish tissue? And so at this time, we believe that there isn't anyone else looking at UV filters in fish. And so we actually have to develop the methods of how we're going to extract those chemical compounds from the fish tissue. And then recently we received an Impact 100 Martin grant, where we're going to be able to look at over 150 fish samples for a whole panel of pharmaceuticals and controlled substances. This particular study will mainly focus on fish from Martin County, but we're going to be seeking funding to be able to look at other fish from other counties as well. Now, this list is always growing, as I mentioned. And so if there are other analytes or pollutants or chemicals of emergent concern that we have the ability to test for, then we can add to this list at any time. So there are 4 major ways for people to get involved in this project. One is that you can help assist us in collecting angler surveys. These surveys are so important because it allows us to really pinpoint what species we want to target. So we don't want people just catching and donating any species for the sake of catching fish. We really want to be very specific and targeted and thoughtful and making sure that we are acquiring fish donations that are coming from these surveys that indicate what species people are consuming. They also are very valuable because they give us that human health component of this project. They tell us how much of a serving are people eating? Who are they sharing that fish with? Are they sharing it with their elderly parents, their young children? Are they sharing it with their neighbors? And then it also lets us know where are they getting their information from, as well as where are the fish actually coming from that they are consuming. So the surveys are very important, and it's important that we're being representative of the population of people that are actually consuming fish. So we're always looking for people to help us with that. Another way is that if you like to fish or get out on the water, you catch and donate fish for the project. They do need to be legal, in season, and kept whole and on ice. And we need to know who caught them, what date, and where you caught them. If you're a high school student and you need to acquire service hours, you can actually acquire service hours for catching and donating fish. I mean, that seems pretty ideal to me if I'm already doing that anyway for fun. Another way is that you can come on into our Center for Citizen Science in Vero Beach and help us process these fish. That means that you're basically taking the tissues and organs that are analyzed out of those fish, and you're doing it in a very specific way. Or you can join us at one of our pop-up events. Our next one is in November at the Environmental Studies Center. And then occasionally we'll have them at other locations as well. And then lastly, you can even learn how to do some of the extraction techniques or protocols to help us extract some of those toxins, intoxicants, or pollutants from the fish tissue. Now, I've kind of alluded to this along the way so far, but we really are focusing primarily on the fish fillet or the tissue that people are consuming, so the muscle. We do reserve the liver and the stomach and the gills and the skin, but at this moment in time, we're really focusing our funding as well as our efforts at looking at what are the contaminants found in these fillets. We've been very fortunate enough to have fairly nice participation in this project from the community. And we've had over 300 citizen scientists engaged in the One Health Fish Monitoring Project. And again, I think that's because there's lots of ways that people can get involved. You can catch and donate those fish to us, where we've had nearly 100 different anglers. You can help us process those fish. So again, come on in and help remove those tissues and organs that are later analyzed. We've trained nearly 40 individuals on how to extract those pollutants, and we have about 50 fisher surveyors who help us collect that information. But we're always looking to expand and increase that number. Now, if you happen to be someone who likes to go out on the water, or you know someone who does, then this is the list of fish that currently have been listed on our angler surveys as being specimens or species that people do in fact consume. So we have a brackish water or marine portion of that survey here, and then we have our freshwater fish. If you see something on here that you like to consume that is not listed, please let me know so we can add that to our list and be a bit more comprehensive. And we are specifically, and you'll see this later on in the talk, that we are really looking for fish from Martin County waters. This is a high priority for us. We do not have appropriate representation, and we are still looking for Martin County fish. So if you are a resident there or occasionally go down there to fish, we need your help. And to make it as easy as possible, what we have done is we have partnered with several bait and tackle shops from Volusia County all the way down to Martin County who have agreed to accept our fish or your fish. So you can go into those bait and tackle shops, drop off those fish, and they'll let us know that they have them, and we can go down and pick them up. So you don't have to drive all the way up to Vero Beach if you're not local. So since our last findings to date, I believe that we have added Mrs. Peters Smokehouse. So hopefully that is helpful for some of you. So at this time, I'd really like to be able to share some of our preliminary results. They're preliminary up into a point where we feel like we have an appropriate sample size, at which point then we can start publishing or sharing more widely. So at this point, they're somewhat still preliminary, but they're still valuable and important. So just to kind of show you where we are with our fish representation, we typically divide the lagoon up into two regions. So we have the northern region, the central region, and the southern region. And so the northern region is composed of Volusia County and Brevard County. Central is Indian River and St. Lucie, and southern are Martin County. So you can see that we have pretty good representation from the northern lagoon. We have 472 fish. We have 733 fish from the central region and a little over 100 from the southern region. Most of the time when we start analyzing our data, we are doing that with respect to region or by county. And so this is what we have with regards to total fish per county. So we have about 1,310 fish total. Only 11 are from Volusia County. We have increased that since the last findings to date, but not by much. We've got a little over 460 from Brevard County, almost 560 from Indian River, 174 from St. Lucie, and a little over, again, 105 from Martin County. So we're getting there, but we really need to bump up our sample sizes for Volusia as well as Martin County. And then what we really would love to be able to do and get to the point of is being able to start localizing some of our counties. So what you are looking at here is a sector map. And specifically, this is Brevard County. And so we have divided Brevard County up into several sectors. That's why you see these color blocks. And what we are able to do is that when someone donates a fish to the project, we know where they caught it, and then we can assign it to the appropriate sector of where it was caught from. And so when we have appropriate distribution or sample size from a particular sector, we can start sharing that information. So let's say that you live near the Patrick Air Force Base, you regularly catch and eat fish from that waterway, then we could hypothetically show you what the pollutant accumulation levels are in that respective sector. So that will be helpful as our project grows. So the first thing I really want to share with you are our microplastic data. I haven't shared with this particular pollutant with you in a really long time. We've worked very hard to increase the number of samples that we've analyzed, and so I'm excited to share that with you now. So first and foremost, what are microplastics? So it's fairly obvious, you know, micro means small. And so we are looking at small pieces of plastic that are categorized as being less than 5 millimeters. And we know through previous literature that these microplastics can be harmful to our oceans and our aquatic life. We also know through the ample amount of literature and studies done on microplastic that at this point, microplastics are ubiquitous in our environment, in our animals, and in our own bodies. And unfortunately, we know that there are human health impacts associated with microplastics. So there's DNA damage, changes to our gene activity, and known risks for cancer development. And also what we know is that these microplastics bind to pollutants, and therefore they act as vehicles throughout an organism's life. And so, you know, these microplastics are essentially bringing these bound pollutants throughout one's body, whether it's in the fillet of a fish, in its stomach, in its blood, brain, or elsewhere. And so the way that we acquire our microplastics data for this study is it's very much citizen science driven. And so our fish samples are, again, donated to the project. They are processed by volunteers, and then additional volunteers will homogenize them. That means essentially they will grind up those fish fillets. And then there's a series of digestions that the citizen scientists will do, and then they will filter it. And then we even have volunteers count those microplastics. So these are two of our more regular volunteers, Sherry and Ellen, who come in weekly to help us start digesting those microplastics. And so we look for microplastics in the stomach of the fish, as well as in the actual fillet or the tissue or muscle. And we can look at microplastics in a couple of ways. We can analyze it by the location that the fish was caught, by the microplastic type, microplastic color, or even the size of that microplastic. So when someone counts those microplastics, these are the categories in which they do so. So let me explain that a little bit better. So first, let's take a step back. When we look at presence and absence of microplastics, we can look at, again, in the stomach or in the fish tissue. So what we have found is of the 428 fish stomachs that have been analyzed for fish stomachs, 71% of those stomachs have some kind of presence of microplastics. And that ranges from 0 pieces to 69 pieces in a single stomach. When we look at presence and absence of microplastics in all fish tissue, we've analyzed 540 individual fish in their fish tissue. 60% of all that tissue shows at least 1 piece of microplastic. And that range so far goes from 0 pieces to 14 pieces in a specimen. And we're looking at a 5-gram portion of tissue. What we also look for when we look for microplastics is we will categorize it by the type of microplastic. And so what I mean by that is if you think about all the sources of microplastic, we have clothing, which is a source of microplastic. We have tarps or sails. We have pieces of water bottles or food wrapping, and all of those sources are kind of indicative of the type. So fiber, for example, is one type of microplastic, and that is represented in picture A and B. So you can see that we have a blue fiber here and we have a red fiber, as well as another blue fiber here. Film, which is kind of pictured in C, D, and E, these are really coming from products like food wrappers or packaging containers, plastic wrapping containers. We have fragments, which is represented by picture F, and that means just kind of a smaller piece of plastic, whether it would be from a plastic pail, a water bottle, a plastic bottle in general. That would be a fragment as it starts breaking down from water, wind, and heat. And so when we look at all the microplastic that we have counted in fish tissue, we find that the majority of that is coming from fiber. So things like clothing, sails, tarps. Then it's followed by fragments, and then film, and then granule. Granules are kind of just little, not beads, but they kind of look the way that they sound, just kind of roundish pieces of plastic. And when we look at that by the counties, this is Brevard County, Indian River County, St. Lucie, and Martin. You can see quickly that blue is the most abundant. And again, that's fiber, and that's being represented by the average here, which is on the picture of the pie chart on the left. So we certainly have the majority of being fibers. And this also jives with current UCF studies, which is looking at microplastics in water. And so again, if that's what they're really finding in the water column, then it does make sense that these fish who are swimming in the water column may be accumulating the same abundance in their bodies. We also look at microplastics by color. So when the citizen scientist or ORCA staff member is reading one of those fibers, not only are they trying to figure out what type it is, but they're also documenting the color. And so when we look at color of microplastics in fish tissue, what we're looking at here is a pie chart depicting the average. So again, this is 540 species or fish that we have tested for microplastics. What we're seeing clearly is the colors blue and black are the most abundant in fish tissue for microplastic. We do see a decent amount of red as well as green, and then the gray portion of the pie chart is other, and that can include colors like pink and yellow and transparent. But again, blue and black are the most abundant. And again, that's represented by our county data, Brevard, Indian River, St. Lucie, and Martin, with those two colors. If we move on to glyphosate, I have shared this data pretty much religiously every 6 months when we do have a findings to date, because as we add more data, this information is changing slightly, and that's to be expected. So I just want to keep kind of updating you as we update our data. So what is glyphosate? So again, glyphosate is a chemical compound. It is typically the most commonly used chemical compound in herbicides. So these are broadleaf products that are used to kill plants. And there is conflicting human health information about the impact of glyphosate on human health, meaning that the EPA's summary review of glyphosate says that it's unlikely to be a human carcinogen. The World Health Organization says that it's probably carcinogenic to humans. And then through ORCA's research, whether it's through this study or whether it's through our pollution mapping citizen science or our land to sea monitoring, a day in the life, or even our larger ecotoxicity studies, we certainly know that glyphosate is persistent in water samples and sediment samples and fish tissue. And there are numerous studies that have tried to look at the impact of glyphosate on fish biology. Most of those studies have been in situ, meaning they're in lab-based or tank studies. So we don't have a lot of information with regards to its impact in the real world. But again, we know it's in the water, and we know it's in the sediment, and we know it's accumulating in fish tissue. So let me share with you how we get that information. Again, just like microplastics, fish specimens are donated to the project. Volunteers will grind the fish fillet up to make it uniform. We will have volunteers extract glyphosate from the fillet using a formic acid extraction technique. And then ORCA staff will run an enzyme-linked immunosorbent assay, or an ELISA, to actually generate the concentration. We have increased the number of fish tested for glyphosate from the last findings to date by over 100. And so now we have over 500 fish analyzed for glyphosate. 99% of those fish have detectable concentrations, and it ranges from below the level of detection, which is 0.05 parts per billion, to just under 1 part per billion in our fish. So what does that really mean? Well, if we break it down by region of the lagoon again, what that means is that we're looking at all fish, all species, and we are generating the average glyphosate of all those fish by the region of the lagoon. So what we're finding at this time is that we have our regions on the Y, glyphosate concentration on the X, and we find that fish from the northern region of the lagoon, so Volusia and Brevard County, have significantly higher concentrations of glyphosate in their fish tissue compared to the central and southern regions of the lagoon. This was done through an ANOVA and Tukey for multiple comparisons, and that is what has generated that significance in this portion of the presentation. When we break this down a little bit more localized at the county level, this is now what we're finding. So we're seeing a decrease from the northern portion of the lagoon, Volusia, and it decreases as we make our way down south to Martin County. And again, this is looking at all fish, all species, and averaging it from the respective county. So for Volusia, you'll note we only have 8 that have been analyzed for glyphosate. So that certainly is not a number that is publishable or really at a point where we can be shouting it from the rooftops. We need to increase that number. And so this was not included in my statistical analysis for this slide, but the other counties are. And so what we have found here is that Brevard County has significantly higher concentrations of glyphosate compared to St. Lucie County as well as Martin County. And so that is one significant difference that we're starting to see. This is different than what we had seen in previous findings to date. And again, this was done through an ANOVA followed by a Tukey for multiple comparisons for average glyphosate concentrations. And then I mentioned sectors in the beginning of this presentation. And so I don't have a lot of sector data to show you, but I do want to just kind of show you what we're working towards. So again, this is just focusing in on Brevard County right now. And we have enough fish from these 5 sectors to be able to share it with you. So again, now what we're looking at is glyphosate in parts per billion on the Y, and we're looking at 5 different average glyphosate concentrations per sector. So this is the average glyphosate for all the fish from BC 19. And so if I go to my map and I look at BC 19, that's this little sliver right here. That's just north of the 404. We have 14 individual species of fish that we could include in this average. Then we have BC 25, where we have 50 fish. So that's the southern portion of Brevard near Grant and Valcaria of the lagoon. BC 13, which is right here, it's the northern Banana River. BC 8 is the northernmost portion of Brevard County's IRL. And then BC 15, which is right here, which is the Banana River between the 528 and the 520. And so we only chose sectors where we had at least a minimum of 10 individual species to be able to average. And so that's what this little n equals. So n equals 14, 50, 33, 21, and 35. And so running statistics on that, what we found is that BC 15, so that is right here, those fish have significantly higher concentrations of glyphosate compared to BC 19. Why is that? Not really sure. But what this does is give us a little bit more of the ability to hone in or localize what we're seeing with regards to the accumulation of pollutants in fish from specific sectors. And I didn't really mention it, but this orange bar here is the average glyphosate concentration for Brevard County. So, you can see that some sectors are below that, while others are clearly above. And all of this glyphosate data was done with the help of funding from the Indian River Lagoon National Estuary Program Small Grant. And so that grant was less than $5,000, and we were able to really springboard this incredible project from all of that, just this portion of our project. And then the next kind of way that we look at glyphosate is we always think about it, well, does glyphosate concentrations vary by different species? And so that's another research question embedded within this. And so that's what this graph is looking at here, or this figure. So what we're now looking at is individual species on the Y, where we have at least 10 individual species, and glyphosate concentrations, again, in parts per billion. And so each of these bar lines are showing you that, yes, different species accumulate glyphosate at different concentrations. So we have black drum having the largest concentration of glyphosate at 0.3 parts per billion, and our hardhead catfish, as with regards to fish from the lagoon, having the lowest average glyphosate. And we've even started to be able to accumulate enough freshwater fish from some of those lakes and canals to be able to start showing you some of that glyphosate data. So we have 2 species of freshwater fish, the Mayan cichlid and black crappie, where we have at least 10 species that we've been able to analyze. And so you can kind of see that at this point in time, those freshwater species have lower average glyphosate concentrations compared to our lagoon or brackish water species. We also are starting to see some significant differences here. So now we're starting to see that, yes, indeed, different species are at a level where they're statistically different than others. And so specifically, we're seeing that the black drum has significantly higher concentrations of glyphosate compared to our hardhead catfish. And we're also seeing that spotted sea trout have significantly higher concentrations compared to the hardhead catfish and the black crappie. Now, this is important, because again, this is a One Health project. We want to know, how is this data impacting or potentially impacting human health? And so we are going to compare our data to established advisory levels when possible. So there are established consumption guidelines for some of these pollutants. So there are some for glyphosate, mercury, microcystin. There are not current consumption guidelines for things like PFAS or pharmaceuticals and controlled substances or microplastics. But when we can compare it to advisory levels, we will. Now, an advisory level, a consumption advisory, is a recommendation. And it's a recommendation to limit or avoid eating certain species of fish caught in water bodies or specific types of water bodies like lakes, rivers, or coastal waters due to some form of contamination. And these advisories may be issued for the general public or for a specific group of people at risk, such as people who eat a lot of fish, like subsistence fishers, which we certainly have a lot of along the Indian River Lagoon, elderly men and women, pregnant women, nursing mothers, and children. So again, there is a current acceptable daily intake for glyphosate in fish. So let's see how our data is comparing. So our highest level of glyphosate in fish comes from the black drum at 0.3 parts per billion. Now, in order to compare that to the acceptable daily intake established for glyphosate, we need to do a little math and convert that to milligrams per kilogram. And when we do that, we get 0.0003 milligrams per kilogram of glyphosate in black drum on average. Then what we need to do is we need to figure out, well, how much does a typical person consume when they're eating fish? And from our angler surveys, we know that the majority of people are eating 8 ounces of fish when they are eating fish. And so if this person here eats an 8-ounce serving of black drum, he would be consuming 0.00007 milligrams of glyphosate in his 8-ounce serving. Now, is that a lot? Well, again, we need to do a little bit more math. So we need to know how much Joe here weighs, which let's say he is an average adult male, so 180 pounds. And now we need to compare this to that established acceptable intake level for glyphosate and its metabolites for people. That varies depending on where you live. So in the United States, that ADI is 1 milligram of glyphosate of body weight per day. In Australia and New Zealand, it's 0.3 milligrams of glyphosate per kilogram of body weight per day. Let's say we live in Australia, they're more conservative. That would mean that it would be acceptable for me to consume 24.5 milligrams of glyphosate. If we go back and we know that an 8-ounce serving of black drum has 0.00007 milligrams of glyphosate, then in order to reach that 24.5 milligrams of glyphosate, which is acceptable, I would need to eat 350,000 servings of that black drum per day to meet that. Clearly, that's not happening, but recognize that our food is just one exposure rate for glyphosate to us. So if I'm eating other food sources that may have glyphosate, like cereals or oatmeal or breadcrumbs or bread or other meats, that would be other exposure rates. If I am working in the industry where I may be exposed to glyphosate, or it's in my drinking water, all of that adds to this cumulative exposure to this particular pollutant. So at this time, to summarize our glyphosate, it's the same as the last 6 months where we shared findings to date. We are not finding concentrations of glyphosate in fish that are meeting or exceeding the current advisory level, which is the good thing. It's not good that it's in our waters, and it's certainly not good that it's in our fish, but it's not at a level of concern. If we move on to look at some of our heavy metal data, then I'd love to do that. So in the past, we have looked at cadmium, lead, and we currently look at mercury. Again, the way that we get that is these samples are homogenized by citizen scientists, and then they are aliquoted, meaning that a volunteer will measure out 5 grams of that ground up tissue, put it in a bottle, and then we send it to our contract lab, Pace Analytics. They then measure heavy metals in our fish tissue. We have previously tested 100 fish for cadmium. Only 2 of those fish came back with detectable concentrations, and therefore we no longer utilize our funding or our resources to analyze cadmium. So that is a positive thing that came out of this study at this point in time is cadmium, yes, it was in our environment historically. It is not entering into our marine food web, into our fish at a level of concern. Positive. Mercury, however, not the same story, and that is why we continue to measure total mercury in all of our fish tissue. We have now analyzed over 1,200 fish species or specimens for total mercury. Again, 99% of those fish have come back with detectable concentrations, and it ranges from below the level of detection of 0.01 milligrams per kilogram to 1.8 milligrams per kilogram of total mercury. The fish that have the highest concentrations, those top 10, well, 8 of them seem to be coming from hardhead catfish and gaff topsail catfish, 1 ladyfish, and 1 crevalle jack. So those top 10 are kind of coming from mainly, it appears, from specific hardhead catfish. Where is mercury coming from? Well, mercury in the environment of Florida is overwhelmingly coming from atmospheric deposition. And that means that the activities across the globe are emitting mercury into the air, where that mercury is then transported in the atmosphere before it ultimately is deposited into Florida's land and waterways and eventually into the marine food web. We know that mercury can affect fish, so it can lead to emaciation, brain lesions, cataracts, changes in behavioral disorders. And we know that that is the same for humans and that it can cause human health implications when we have too much of it. And so that is, again, why we track it. And so when we look at average mercury in fish per county. Again, we are looking at all species, all fish averaged together from that specific county. So that is what you are seeing on the X is the 5 different counties. The Y is average mercury in milligrams per kilogram or parts per million. You can see that it changes slightly depending on what county you're in, but obviously, again, note that we have a very small sample size from Volusia and we have a relatively small sample size for Martin County. We'd love to see this number bump up to at least 105, 120 or more. The purple line at this point is the average for all counties. And so what we're finding at this point in time is that, again, fish from Martin County have significantly higher concentrations of mercury compared to all the other counties. And we're also finding that Brevard County and St. Lucie County have significantly higher concentrations than Indian River County. Again, this was done through an ANOVA and a Tukey post hoc test for multiple comparisons. The same question is asked about, well, how does this information relate to current advisory levels? So for this particular figure, what you're looking at are the established advisory consumption guidelines for mercury in fish tissue. And so the orange line is representing the Florida Department of Health advisory level for women of childbearing age and children. The green line is a joint advisory level from the EPA, Florida Department of Health, and FDA for the general population. You can clearly see that on average, mercury concentrations of fish from all counties exceed the FDOH advisory level for women of childbearing age and children, and that Martin County fish meet that advisory level established by the several agencies. Our standard error bars indicates that there are some fish that exceed that general population advisory level, but on average, Martin County fish meet that joint advisory for general population. Even with adding several new fish to Martin County and all the other fish since the last findings to date, we're still seeing that same trend. Same question about glyphosate. Well, does it matter if you're looking at different species? Does average mercury accumulate differently by species? And again, our data is indicating that yes, it does. Now, that's not all that surprising. I mean, there are, again, established mercury guidelines for fish. I mean, usually we're talking about fish that are higher up in the food chain, like tuna and swordfish and those larger fish. But when we look at species from the Indian River Lagoon and those freshwater canals, we are also seeing differences in mercury concentration accumulation. And so we are now at the point where we can show you 17 different species where we have at least 10 species that we've been able to test for mercury and then average that. So again, we have 11 striped mullet, 19 Atlantic croaker, 240 mangrove snapper. And so, you can see it varies a bit, but this figure is showing you species where we have at least 10 data points to create that average. That doesn't mean that we don't have other species, we just don't have at least 10. So it goes from lowest average mercury to highest. And so we are seeing that striped mullet, croaker, and black drum are kind of lower on this figure, whereas our bluefish, crevalle jack, and ladyfish are our fish species that have higher accumulation concentrations. Once again, we've overlaid those advisory levels. So the orange line, again, is representing the Florida Department of Health advisory for women of childbearing age and children. So we do see several species that do not meet that advisory level. Black crappie, bluegill, Mayan cichlid, those are our freshwater species. Black drum, croaker, and striped mullet. And then we do see several species that exceed that joint advisory level for the general population. So that includes our spotted sea trout, ladyfish, crevalle jack, and bluefish. And so, this figure at this point is indicating that different species do accumulate mercury concentrations at different amounts. And it also can help individuals who do consume fish from our lagoon perhaps decipher what species they would like to target if they are in fact going to consume those individual fish. So it just gives people a little bit more information, data to make informed decisions. And then again, I just wanted to highlight that we're at a point where we're starting to really be able to include some of those freshwater fish, and that's what these green bars are. I've kind of already alluded to that, but just highlighting it in this figure here, that's exciting because there are, again, people who specifically eat fish from our canals and freshwater lakes. So somewhat lower in overall mercury concentration, but there are several species within each bluegill, black crappie, Mayan cichlid that exceed that FDOH advisory level, but not all. So those are really the three major analytes or pollutants that I'm able to talk about tonight. So that was microplastics, glyphosate, and total mercury. Again, we have data on PFAS or forever chemicals that we can also share, cadmium as well as lead. So if that is of interest to you, you are free to reach out and I can share that information with you. And as soon as we have pharmaceuticals and controlled substances as well as UV data, I'll certainly be sharing that with you at the next findings to date. But as far as the data that was presented at this particular presentation, what we know is that blue microplastics are the most common color. We know that fiber is the most common microplastic type. This jives with current UCF research looking at microplastics in water column. And so it's not all that surprising to now be able to document that in fish from the Indian River Lagoon. We know that glyphosate is ubiquitously found in fish. 99% of all of our fish, over 500 individuals have been tested, have detectable concentrations, but those concentrations are very low and below acceptable daily intake advisory levels. We also know that mercury concentrations vary geographically and also by species. And we know that in some cases, certain species are accumulating mercury at concentrations that are exceeding current advisory levels for human health. I have made a call, I continue to make the call for action that we need more fish from Martin County and Volusia County to be able to drive these this data more concurrently and to be more positive and confident in that data, but what we have right now is preliminary. So as we look ahead, we're going to focus our efforts on acquiring fish from Martin and Volusia County. We are welcoming anybody who would like to join us at our next pop-up event in Martin County at the Environmental Studies Center on November 13th. This is a chance for you to learn how to process fish. You can also drop off your fish that you may have caught there or caught from Martin County or St. Lucie County while I'm there. But we often host processing events at our Center for Citizen Science in Vero Beach about once a week or a couple times a month. And so you can also learn how to do that at that point. I'm really excited to be able to share with you what we end up figuring out with our UV filters, or again, these are the compounds found in sunscreens. And so we're working with our colleague at Embry-Riddle to develop those methods of how to detect that. And then I'm very much looking forward to being able to share with you the pharmaceuticals and controlled substance data that we will acquire from Clemson University, really focusing primarily on fish from Martin County at this time. This is just a community ask that if you are interested in collecting fish for us, this is what is required of you. I can go over that in more detail if you'd like to. My email address is also here. So if you do want to reach out to me about this project in any capacity, or you'd like to get involved, or you have questions about the data I presented tonight, please feel free to reach out. I try to answer your questions in a timely way, but it's mweiss@teamorca.org. I definitely would like to acknowledge and thank all of our incredible volunteers. Many of you are on the call right now. thank you for all of your time and your effort. You can choose to volunteer with any organization, but you choose to give some of your precious time to us and to help us in our research. And we are so thankful of that. I'd love to thank my colleagues, as well as all of our ORCA interns who have worked on this project, and then our two college collaborators at Embry-Riddle and University of Florida for their assistance on current research. And of course, our funders, because we can't do any of it without money, and science is expensive. So thank you to all of those who saw the importance in this study and the importance it has for our community. So again, thank you. With that, I would love to take any questions that you may have. You may unmute yourself or put it in the chat and I can try to answer them for you. Or if you'd rather just reach out to me privately, feel free to email me, stop by our Center for Citizen Science, or give me a call. But thank you again for being here. And if you have any questions, I will take them now.
Hey, Missy, it's Bonnie. How are you?
Good. How are you?
Good. Thank you for the data. I just had a quick question. Why analyze or why share the data on the color of the microplastics? Is there something additional research there, or?
Kind of. So sometimes what that allows us to do is to start thinking about the source. So what I didn't share is that not only is blue the most common and fiber is the most common, but blue fibers are actually the most common. And if we think about what are sources of blue fibers in and around the water, can you think of anything where that might be?
Well, hopefully not our blue ORCA t-shirts, but
I hope not as well. But what I'm really thinking here we're thinking about tarps. So if you think about most tarps that are covering our boats, are covering or waterproofing our kayaks or whatnot, a lot of them are blue and they are producing a lot of the microplastic that is entering into our waterways. So it kind of goes back to the source of what that might be.
Okay, gotcha. Thank you.
Well, I will leave it one more time by just thanking you for being here and thank you for spending your evening, your dinner time with me, talking about fish and fish tissues and organs. I'm sure it's incredibly appetizing. But again, it takes an army and it takes a village. And even though sometimes this information can be depressing and dire, it's really important. And it allows us to provide the ammunition, the evidence, the data to share with decision makers and stakeholders and the community to try to reverse some of the degradation that our lagoon is impacted by. So it's not just important for information, it's important really for animal health, human health, and the fight for clean water. So again, thank you for being here. If you're interested in getting involved in the project, we would love to have you. Feel free to email me. And to everyone else, I hope to see you very, very soon. So thank you.